Welcome to part 3 of chapter 1. This presentation will talk about the modes of transmission of parasites. For the outline of the presentation, the first part of the presentation will talk about the introduction to the different routes of transmission, and we will also talk about the different routes of parasite transmission with their examples. For the learning objectives, this presentation aims to describe the different routes of transmission of parasites between hosts and cite some examples of parasites that exhibit specific route or routes of transmission. For the introduction to the routes of transmission, the parasites are commonly transmitted through ingestion of parasite-contaminated food and water, besides other routes such as skin penetration, intranasal transmission, coitus, and congenital transmission. Each mode of transmission will be dealt with in detail with suitable examples. Parasites have distinct routes of entering a host. Endoparasites enter the host in general through natural openings on the exterior of the body or through the skin. Parasites are transmitted from one animal to another by ingestion, skin penetration, contact, predation, coitus, and transplacental or transmammary transmission. The first mode of transmission that we are going to discuss is the transmission through ingestion. The parasite transmission through ingestion is considered to be the most common mode of transmission of parasites. The host may become infected by direct ingestion of an infective stage through contamination of food and water, or by ingestion of an intermediate host or transport host containing infective stages. This diagram shows the mode of transmission of parasitic gastroenteritis in ruminants. As you can see here, the survival stage of the L3 or the third larval stage of the parasite happens in the environment. The infective stages or the third larval stage may climb up the vegetations and wait for the host to ingest it. The infective larvae are present in the grass blades. The ingestion of the infective L3 represents the major mode of transmission whereby parasitic gastroenteritis in ruminants will occur. This is especially true for GI nematodes. Another parasite that exhibits transmission through ingestion is the parasite that causes coccidiosis in dogs. So when we say coccidiosis, now that is the clinical illness caused by infection with the protozoan parasite of the genus Eimeria. So these particular parasite are collectively known as coccidia. So the genus of coccidia that infects dogs and causes canine coccidiosis is the genus Isospora. So in this diagram, we have here the presence of the oocyst in the feces now coming from the infected dog, and the oocyst later becomes a sporulated oocyst that will contaminate the food or the drink, and it can later become ingested by the canine host. So dogs again become infected with coccidia from ingestion of the oocyst that are found in the dog feces and soil contaminated with feces. Another example of parasite transmission through ingestion is exhibited by the parasite that causes poultry coccidiosis. So chickens become infected by ingesting the sporulated oocyst or coccidian eggs from the surrounding environment. So these coccidian eggs is from the genus Eimeria. Another parasite that exhibits transmission through ingestion is the Ascaris suum. So the main host of the Ascaris suum is the pigs. So Ascaris suum is also known as the large roundworm of pigs. The pigs become infected by the parasite through ingestion of the embryonated eggs that are present in the environment. For parasites with indirect life cycle, the host becomes infected by ingestion of an intermediate host or transport host containing the infective stages. When we say a parasite with an indirect life cycle means that that particular parasite requires an intermediate host to reproduce. Parasite that exhibits this characteristic is the Dipylidium caninum. Dipylidium caninum is a common tapeworm of dogs and cats and occasionally humans. As you can see here, the intermediate host of the parasite Dipylidium caninum is the flea, the cat or the dog flea of the Tenocephalidis species. The flea harbors the cysticercoid form of the parasite, Dipylidium caninum, as well as its larval stages until the parasite becomes mature. The vertebrate host, in this case the dog, becomes infected by ingesting the adult flea containing the cysticercoid form of the Dipylidium caninum. So this is an example now of a parasite that is transmitted by ingestion of the intermediate host now that contains the infective stages of the parasite. In this case, now the parasite is the common tapeworm Dipylidium caninum. In certain parasites, the definitive hosts are infected through ingestion of raw or undercooked meat, organs, or milk of intermediate host. So these are exhibited by the cystodes or the tapeworm Taenia solium and Echinococcus granulosus. So an example of a parasite that exhibits this particular mode of transmission is the Taenia saginata. So Taenia saginata is considered to be a helminth of the cystode group. Taenia saginata is also known as the beef tapeworm. It is a zoonotic tapeworm. It is an intestinal parasite in humans, causing taeniasis and cysticercosis in cattle. So the cattle serve as the intermediate host of the parasite, harboring the tissue cysticerci in their muscle, while the humans are considered to be the obligate definitive host, harboring the tapeworm form. So this is an example of a parasite that is transmitted to humans by ingestion of raw or the undercooked beef that is infected with the larval stage of the Taenia saginata, which is the Cysticercus bovis. The next mode of transmission is through skin penetration. So after mouth, the skin is considered to be the most common route of entry for many parasites. The infective stages of the parasite may actively penetrate the skin, and in this process, the secretions of the parasite help to digest the host tissue. An example of a parasite that exhibits skin penetration or that is transmitted through skin penetration is the Cercariae of the cystosomes. So we have here the diagram showing the life cycle of the schistosomiasis or the bilharziasis. Schistosomiasis is caused by some species of blood trematodes or flukes in the genus Schistosoma. So there are three main species infecting humans, and these are the Schistosoma hematobium, japonicum, and the mansuni. The definitive host of these parasites are humans. Transmission occurs when the cercariae is considered to be the infective stage of the parasite. So this is in this life cycle, this is shown here at number 5. The transmission occurs when the infective cercariae swim and penetrate the skin of the human host. So this is an example of a parasite now that is transmitted by way of skin penetration. We also have the intestinal hookworm. So the intestinal hookworm disease in humans is caused by the helminth parasite Ancylostoma duodenale, Selanicum, and the Necator americanus. So this is the life cycle of the intestinal hookworm. Now showing the number 1 here shows the eggs of the parasite that is present in the feces. The eggs later become the rhabdidiform larva. The rhabdidiform larva later develops to filariform larva in the environment. Later, the filariform larva will penetrate the skin of the host, not particularly humans. So at number 4, the infective larvae, which is the filariform larvae, on contact with the human host, typically a bare feet host, the larvae will penetrate the skin and are carried through the blood vessels to the heart and then to the lungs. They penetrate into the pulmonary alveoli, ascend the bronchial tree to the pharynx, and are swallowed. So this particular parasite, now the intestinal hookworm of the genus Ancylostoma, is an example of the parasite now that is transmitted to its host by way of skin penetration. Certain parasites enter the host via the bite of an intermediate host serving as a vector. These parasites develop in blood-sucking insects and ticks, and when these vectors feed to obtain the blood and tissue fluids from the host, the infective stages of the parasites are introduced into the host. So an example of a blood-sucking parasite now that is responsible for the transmission of different types of parasites to its host are the ticks. Ticks are considered to be blood-sucking parasites that attach themselves to animals and people. Once attached to a host, the ticks will feed voraciously. As they feed, the ticks can transmit a large number of diseases, including the Rocky Mountain spotted fever, ehrlichiosis, the Q fever, and Lyme disease. This diagram shows the typical life cycle of brown dog ticks. Brown dog ticks spread the bacteria that causes Rocky Mountain spotted fever in some parts of southwestern US and Mexico. The Rocky Mountain spotted fever is a bacterial disease spread through the bite of an infected tick. Most people who get sick with RMSF, Rocky Mountain spotted fever, will have fever, headache, and rash. So this is an example of a transmission that is made through a parasite. Now, and that parasite is the bacteria that causes the Rocky Mountain spotted fever. The bacteria is being harbored by the tick, a blood-sucking parasite of the, particularly the brown dog ticks. Another type of transmission is by contact or direct contact and predation. Parasites are transmitted between animals by contact, especially when they are confined in sheds or houses. Example of a parasite that exhibits this particular type of transmission, direct contact, is exhibited by lice. Lice infestation and mange are chiefly transmitted between animals by contact or direct contact. So we have here an example of a lice, the adult little blue cattle louse, Solenipotes capillatus. Lice are blood-feeding or skin, hair, feather chewing ectoparasites that are a menace to pets, livestock, and humans. Lice are transferred between hosts by direct contact, by crawling, or by foracy, which means they hitch a ride on another insect in order to find a new host. Lice are transmitted from one animal to another by direct contact. Up to 1 or 2% of the cattle in a herd can carry a high load of lice, even in the summer when high temperature reduces the number of lice. So these are examples of lice, all females, that are present in cattle. We also have here examples of poultry louse species. This one here is an example of a chicken body louse. We have here the shaft louse, the fluff louse, the chicken head louse, the Menacanthus cornutus, and the chicken wing louse. So all of these parasites are transmitted between hosts, particularly between poultry species, by way of direct contact. Another group of parasites that causes or that is being transmitted by direct contact is the mites. The mites causes the condition known as mange. Mange is a contagious disease characterized by crusty or scaly skin, pruritus, and alopecia. Mange is a general term for cutaneous acariasis and is a result of infestation with one of several genera of parasitic mites, including Coreoptes, Demodex, Sorobia, Soroptes, Sarcoptes, and others. All of the mites that cause mange are spread by direct contact. The scabies and mange mites are highly contagious, meaning that they are easily spread to other animals or people. Some mites can survive for several days off an animal's body, on clothes, towels, or bedding. So these objects can be a source of infestation. This figure is the figure of a mite, the parasitic mite Sarcoptes scabiei. Sarcoptes scabiei, or the itch mite, is a parasitic mite that burrows into the skin and causes scabies. Parasites are also transmitted during predation by a host. For example, when cats predate on rats, cats may acquire an infection with parasites. An example of this is the Toxoplasma gondii. So we have here the life cycle and infection with Toxoplasma gondii. So the life cycle of Toxoplasma gondii is quite complex and involves two types of host, we have the definitive host and the intermediate host. The definitive host of Toxoplasma gondii include the cat, including the wild cat and the domestic cats. The intermediate host of the Toxoplasma gondii include several other animals. Most cats are infected by eating meat containing Toxoplasma gondii cyst that can include raw or inadequately cooked meat, or more commonly prey species. In this case, we have the rodents that can serve as a prey for cats, now, which are the definitive host of the protozoan parasite Toxoplasma gondii. Another mode of transmission is through entry through nostrils. An example of a parasite that is transmitted through the nostrils are the nasal botfly Estrus ovis. Estrus ovis deposit its young larvae around the nostrils of the host, whereupon they crawl upward and enter the nasal sinuses. So the sheep botfly, or the Estrus ovis, is an obligate parasite, and it cannot complete its life cycle without utilizing the nasal passages, frontal and maxillary cavities, and sinuses of most domesticated sheep. So we have here a diagram showing the life cycle of the Estrus ovis, the nasal botfly. So as you can see here, the life cycle of Estrus ovis is considered to be direct, and the first stage larvae migrate through the nasal passages to the frontal sinus, where they develop into third stage larvae, then migrate back to the nostrils and are expelled into the environment through the host sneezing. The larval development in the nasal passages and the frontal sinuses takes up to 9 weeks. Another example is the parasite Naegleria fowleri. This is a freshwater amoeba present in surface water of polluted pools, gaining access to the host through the intranasal route. Naegleria is an amoeba. Amoeba is a single-celled living organism commonly found in warm fresh water, for example, lakes, rivers, and hot springs, and soil. Only one species of Naegleria infects people, and that is Naegleria fowleri. So this is a diagram showing the life cycle of Naegleria fowleri. Naegleria fowleri infects people when water containing the amoeba enters the body through the nose. This typically occurs when people go swimming or diving in warm, freshwater places like lakes and rivers. The Naegleria fowleri amoeba then travels up the nose to the brain, where it destroys the brain tissue. Naegleria fowleri causes an acute fulminant meningoencephalitis known as the PAM, or the primary amoebic meningoencephalitis, and this is acquired by swimming or water skiing in small, shallow freshwater lakes as a result of direct contamination through the nasal cavity. Naegleria fowleri is colloquially known as the brain-eating amoeba. It is a free-living microscopic amoeba, and it can cause a rare and devastating infection of the brain, which is called as the primary amoebic meningoencephalitis. Another mode of transmission is the entry through external genital. Example of a parasite that exhibits transmission through external genitalia is the Trichomonas fetus. So this parasite is a protozoan parasite that causes abortion in cattle. We also have the Trypanosoma equiperdum, the protozoa that causes dorine in horses, and they are mainly transmitted through coitus. So we have here the trophozoite of the Trichomonas fetus in gem-sustain, and we also have here the parasite, not Trichomonas fetus, in scanning electron micrograph. The parasite, Trichomonas fetus, is an extracellular flagellated protozoa, an organism responsible for sexually transmitted disease, known commonly as bovine trichomoniasis. So we have here the illustration of the life cycle of the Trichomonas fetus. We have here an infected male, and we also have here the infected female. In the infected male, particularly in the bull, the parasite is located in the preputial mucosa, and the female, the parasite is located in the os cervix. Since the parasite is mainly transmitted through coitus, the other term for that particular transmission is also known as the venereal transmission. So venereal transmission is defined as the transmission of the parasite during mating. So the parasite, Trichomonas fetus, is considered to be an obligate parasite of the female reproductive tract and surface of the bovine males, prepuce, and penis. The bull is considered to be an asymptomatic carrier of the Trichomonas fetus organism and may efficiently transmit the protozoa to the female during coitus. Under natural conditions, the parasite is transmitted directly from an infected animal to healthy animal, almost exclusively through natural mating. The bulls become infected during the mating of the infected cows, remaining asymptomatic carriers. We also have another parasite that is transmitted mainly by way of the external genitalia or through coitus, and that is Trypanosoma equiperdum. So Trypanosoma equiperdum is the cause of dorine, and it is considered to be the only parasite, protozoan parasite, known to produce venereal disease in horses. So this diagram here represents the Trypanosoma equiperdum trypomastigotes blood smear at 1000 times magnification. The last is the transplacental or the transmammary transmission. Transplacental or prenatal transmission refers to the transmission of the parasite from mother to fetus across the placenta. So an example of this is the Toxocara canis. We also have the transmammary or the transcolostral transmission, and this refers to the transmission of the parasite from the infected dam to offspring via colostrum or milk. So example of the parasite that exhibits this particular mode of transmission are the helminth parasites, Toxocara vitulorum and Toxocara cati. So an example of the parasite that exhibits transplacental or transmammary routes of transmission is the Toxocara canis. So this diagram here represents the life cycle of Toxocara canis. So the at one part of its life cycle, you can see that there is the transplacental and transmammary transmission of the parasite. And this happens between the dam and the offspring or the puppy. And at this particular phase of the life cycle of the Toxocara canis, the insisted stages that are the insisted stages of the parasite that are present in the dam are reactivated in the female dogs during the late pregnancy. And after the reactivation, during birth or during lactation, the parasite can be transmitted to the puppies. So another example of the parasite that exhibits transmammary transmission is the helminth parasite Toxocara vitellorum. So this is the theoretical life cycle of Toxocara vitellorum in bison, showing that at one point of the life cycle of this particular parasite, there is transmammary transmission, or the transmission of the parasite from the dam to its offspring by way of the mammary gland.